Let's resume from question number 60th because my recording just stopped there. So they were asking about which of the method you cannot use for locus probe analysis of ENTR region. So DNA extraction you'll have to do. So it can't be the answer. B is restriction endonuclearization. As we all know, restriction endonuclease type 2, they are very specific. Therefore, they can help you identify a single locus because they can cut at the specific site. So this can also not be true. C is gel electrophoresis. That is the only method by which you can run your gel and you can get a picture. You can't see DNA and tell that okay fine, this is the region where your as SNPs or VNTRs are present, right? So all three of them they cannot be the answer. So the fourth one, whatever it will be, so the fourth one is recombinant DNA. Yes, you can't use it, so recombinant DNA will be the answer. 61st question says which of the following elements is not a characteristic of factor independent terminators in E. coli. So in E. coli, whatever is factor independent is intrinsic, right? So it is saying that it's C rich sequence. As far as I remember, it's not C rich, it is GC rich. So B is saying that RNA sequence that can form stem loop. Yes, because that is the only method by which it can undergo intrinsic termination by forming stem loop, which cannot be bypassed by the RNA polymerase. So the C option says a run of single stranded U residues. Yes, U residues, U stretch says that it is the position where the your transcription has to halt, right? So D option is GC rich sequence. So all three of them are correct. Therefore, A answer would be right. So 62nd question says a covalently closed circular DNA containing a single promoter is mixed with RNA pole that open complexes form where one turn of DNA is unwound. Which of the following statement will be true? So let me take you to the process. It is the place where your RNA is opened, right? If one turn is opened here, we all know that if it is unwinding here, then there is overwinding here. So what will happen if here it will be -1, then here it will be +1. You can do it by the example of thread, by the example of thick thread. So what will happen if you create a loose stretch here? The thread will be supercoiled at the front or either of the end, right? So it will be supercoiled here, therefore it will be nullified. It is saying that here it is unwounded. So what will happen? So the thing is, it will be nullified. If it is -1 here, then it will be +1 here. It is unwinding here, then it is overwounding at the downstream of the DNA. So the answer will be L stay the same because an increase in width will cancel out the decrease in T. So the option number D will be correct. Then 63rd question is saying that there is RNA from the ribosome of E. coli has GC content of how much? 51%. Now what happens? This E. coli is infected by a bacteriophage. When bacteriophage infects it, the GC content of bacteriophage is 40%. Now let's do what? Run a density gradient. In a density gradient, we get different peaks. So the peaks are, there is 23S, then there is 16S, then there is 4S. But there is saying that there is a baseline peak here. There is a baseline peak here in between 23S and 16S. So they're asking which peak is whose, like of either from E. coli and the bacteriophage, this baseline peak is whose, right? So the first option is saying all the RNA will be 40%. It cannot be possible because there is also this E. coli or the host cell. So host cell will also produce some of its RNA. So all the RNA cannot be from the 40% GC because when the RNA is extracted, E. coli's RNA will also come out. So B is the RNA in the 4S peak will be 40% GC, but everything else will be 51% GC. This cannot be true. Why? Because E. coli is in living condition, and whatever material will be utilized by phage will come from E. coli. So the RNA concentration or the GC in RNA concentration will of E. coli will not decrease. So the thing is 23S, 16S and 4S, all of them will be for 51% GC. Why? Because all the material is coming from what? Is coming from your E. coli. So the thing is, and also 23S, 16S and 4S, it is prokaryotic ribosome, right? Your virus is going to use E. coli's ribosome. So whatever the baseline peaks were, whatever the peaks were will be from E. coli. So they all will be from 51% GC content RNA. So the option number C will be correct. But the material between 60S and 23S will be 40% GC. Okay, now comes 64th question. Which of the following statements about tumor suppressor is true? A says, it says something about tRNA and tumor suppressor has nothing to do with tRNA. B says tumor suppressor are mutated viral versions of cellular proteins involved in cellular transduction. See, this option is fine but is not correct. They are not mutated viral versions. These are our versions, like the viruses came, infected us, okay, the higher organism, and they mutated them, and then they took those viruses, they inserted those small fragments of DNA into their genome. So they are not mutated viral versions, they are mutated like our versions, mutated, what will say it, they are non-viral, mutated non-viral versions. So this option will also be incorrect. Then comes option number C, recessive mutations that inactivate the RB tumor suppressors are found in the families with high incidence of retinoblastomas. This is completely correct, right? Why to read option number 4. So 65th question says that the insulin receptor functions, as we all know that insulin acts as what? RTK, receptor tyrosine kinase. So it functions as tyrosine kinase. Then comes 66, frame shift mutations are observed because the DNA code is, why do we observe frame shift? Because it is comma-less, right? So fine, if I'll insert something from a different color, I can insert it here, right? If at all there was a comma, then the cell could identify the fact that we cannot insert it here because it is the ending of one of the codons. So because it is comma less, therefore frame shift mutations are observed in the nature. Then we come to 67th question, which says when synthetic mRNA consisting of alternating A and C residues were translating C, it is saying AC, AC, AC. When it was translated, then two proteins we got. One was what? One was threonine and the other was histidine, right? So then it is giving you options. From this you can write, it is forming polyasparagine, polythreonine and polyglutamine, right? So it is asking you which of the codon will be from histidine. So this codon will be from histidine. However, I have not solved it in the manner I should, but I want you to solve it because the other problem which they have given, other codon which they have given, if you will superimpose, if you will write that code, say for example, if you write AAC, right, ACA CAA, and you'll write whatever is given like polyasparagine, polythreonine and polyglutamate, aspartate glutamate, right? So what will happen if you will superimpose them, then only one of the codon is left, that is CAC. So answer will be CAC. Now come to 68th question. Hybrid dysgenesis is asymmetrical. It is induced by, see, hybrid dysgenesis only happens when male is not able to repress the transposon. So it is because of P male and M female cross. So the option number B will be correct. 69 says during RNA polymerase second transcriptional initiation, phosphorylation of the following factor is essential to commence transcription. So which of the phosphorylation is essential? Again a theoretical question. It is CTD. C terminal domain has to get phosphorylated. Okay, remember the amino acid stretch which is present at the CTD. 78th question says typical nucleosomal organization of a gene is not found in, see, in all of them except sperm, because spermine and other histone proteins are also present in the sperm, and the typical H2H2BH3H4 core as well as the linker histone, this organization is absent in the human sperm. Then we come to 71 question. It is saying a mutational event. There is a mutational event inserts bases in the beginning of the coding sequence of gene. The highest chance of the altered protein being functional when the number of bases insert. So it is asking you that whatever protein gets inserted in your genetic code, if you want that protein should also be functional, then what is the number of bases that is required for that protein to be functional? That is 3, because only then the inserted protein will be able to form a protein of its own. So it will be 3, right? Because our codon is of how many amino acids? 3 amino acids. Then we come to 72nd question and the last question of this session. This is in a temperature sensitive mutant bacteria at non-permissive temperature, there are huge accumulation of Okazaki fragment. The bacteria are mutant proof. See, if there is an accumulation of Okazaki fragment, then only one fact can be there, that is they are not sealed. If they are not sealed, then the enzyme which is responsible for sealing them is mutated there. So what is that enzyme? That enzyme is DNA ligase, right? So the option number C will be correct. After that, this question is comprehensive kind of question. Then you have to read this question and then solve this question. So from this question, I will do them tomorrow or day after tomorrow, right? So keep studying, and I'm doing what I'm making one more series, that is mock test questions, and in them I will tell you there will be mock test series come short notes. So what I'll do, I'll make one liners, because the format of DBT is like that only. So say for example, what is tripoperon, right? Where is attenuated is located. So with this, I will stop this video here only, and thank you. Keep studying.